Can everyone hear me okay?
Yes.
Okay. All right. So good morning, good afternoon, good evening. Thank you very much for that kind introduction, Christoph. My name is Maggie Couvillon, and as was said, I'm an assistant professor at the Department of Entomology at Virginia Tech, which is a university in Virginia on the East Coast of the United States. So, I actually had a note to say in my introduction exactly what Axel said, which is one of the silver linings of the past 2 years has been these exact opportunities, the chance for us to interact with colleagues and friends worldwide in ways that that give us a little bit of hope for the future. So I'm going to go ahead and jump right in. So today I'm going to talk to you about research that we're doing at Virginia Tech that I hope one day will lead to recommendations to help feed hungry pollinators. So I'll be showing you for the first time, actually, some new data from my lab. So again, thanks for giving me a reason for having to update my talk and for being my guinea pigs. So jumping right in, as you've probably heard, bees are important to our world. So many of the things that add the diversity and the nutrition and the flavor to our diets are there partially or completely thanks to bees. So the almonds and the cranberries, parsley, berries, broccoli, cherries, citrus, these are here thanks to the work of the bees. And of the insect pollinators in general, bees are really our heavy lifters. They dominate agriculture in a way that no other taxa does. So this is data taken from two recent papers where we have number of crops on the y-axis and we have different pollinating taxa on the x-axis, and you can see that the 1, 2, and 3, the top 3 positions are occupied by bees. So as having such an important role, they really carry immense economic clout. In the United States, bees add anywhere between 17 to 20 billion annually in crop value alone to the US economy. As you've also probably heard, bees are declining globally in biomass and abundance and species richness, just about any way that you can measure it. Here we have percentage declines in some different insect groups, with bees that I've circled here in red showing a 46% decrease in the last decade. If we look even just at our managed bees, our honeybees in the United States, the number of managed hives is declining in the US. It's increasing in some parts of the world like China and Argentina, but overall the demand for what we need the honeybees to do is not keeping pace with the supply of them. So in the United States, we reached our peak of managed honeybee hives right after World War II, around 1945, which you can see here. And at the moment, we're kind of down here at about 2.2 million. Now, the reasons for the decline in both managed and wild bees is complex and multifaceted. And those of us in the field usually shorthand it as the four.
Recording in progress.
So we say that bees are troubled by pests, like this mite that's hitching a ride on the back of this bee, pesticides, not just insecticides, but also fungicides and herbicides, pathogens like the virus that causes these shriveled wings, and poor nutrition, where foragers are not able to find abundant and nutritious pollen and nectar in the landscape. These stressors can act singly, but growing evidence is showing that when they act in concert is really when the biggest harm is done. Now, my research focuses on the stressor of poor nutrition, because whether or not your colony is healthy or sick, it still needs to eat. And anthropomorphic landscape changes have reduced the available forage for bees in the landscape, not just because we've paved over to make our cities, but more consequently, because we've plowed under to make our agricultural land. So intensively managed agricultural land like this green field is such a common feature in the rural landscape in many first world countries that it might be difficult to remember that just 60 years ago, 97% of this used to look like this, sometimes referred to as unmanaged prairies or grasslands or hay meadows, but essentially all mounting to the same thing of large swaths of land with abundant wildflowers that are blooming throughout a foraging season. And the good news is people want to help. So there's recently been an upsurge of interest from federal policymakers down to everyday citizens. People want to feed the hungry bees. But especially when we're talking about a landscape level, how can we direct our efforts to make them targeted so that we can most help the bees in a large landscape that's not feeding them as well as it should? And that's where my research comes in. So welcome to the Couvillon lab at Virginia Tech, where we let the bees themselves tell us, with all their hard work, about how good the landscape is doing at feeding them. I started at Virginia Tech in 2017, and I've largely been working on what I'll be talking to you about today, which is we're letting the dancing bees evaluate the landscape for its ability to feed not just them, but also other pollinators. Since I've been at Virginia Tech, we've been investigating this question in 3 different landscapes, and today I'm going to talk to you about 2 of those 3 landscapes. How honeybees are doing in a row crop environment, how honeybees are doing in a fruit, specifically an apple orchard. And then thirdly, if there's a little bit of time left, I want to tell you just some quick cool things that dancing bees can also tell us. So jumping right in, I want to introduce you to the waggle dance. So a forager who has found a good source of food, usually nectar or pollen, returns to the hive, and she communicates the direction and the distance from the hive to the food. So you can see, hopefully the video is playing, a bee dancing here. She is, we're going to go back and play that again. She has her head facing down while she rotates her abdomen very quickly from side to side. Then she stops, returns, repeats the part where she rotates, stops, returns, repeats the part where she rotates, stops, and returns. Now, both of these components, the direction and the distance, are communicated in the part of the dance where she's moving her abdomen very quickly. So for this bee, it's when her head was facing down. We're going to look at that a little more detail, but I want to give you a little bit of history on it. The waggle dance we've known about for thousands of years. Aristotle wrote about it in his work. But the decoding of the waggle dance, the understanding what it was actually communicating, was done thanks to this gentleman, Carl von Frisch, an Austrian ethologist. He shared a Nobel Prize in 1973 for the discovery of the waggle dance. So here's some footage taken from the movie More Than Honey that shows von Frisch doing his seminal experiments. I'm going to back up again and play that video. So here there's a lady who's putting some artificial sugar solution out, and von Frisch is looking at the bees through a glass-walled observation hive using a protractor to measure angles, and wearing lederhosen like all good Austrian ethologists should. So let's break down a little bit what exactly von Frisch discovered for us. How are the bees communicating the distance and the direction? We're going to start first with direction. So here we have a bee in yellow, and she is foraging on a good source of flowers. So these blue ones down at the bottom. She returns to the hive, and before she goes into the hive, she finds where the sun is and drops it to the horizon. And then she measures the angle from where the sun is on the horizon to where the forage is. So in this case, the angle is 180 degrees. Now, if these blue flowers had been over here to the right, the angle that she measured would have been 90 degrees. Then she goes inside the hive and she recreates that angle that she just measured, where the sun stands in for straight up on the comb. So she measured 180 degrees, and she comes inside, and here's the comb. So straight up is 0 degrees. And to make a 180 degree angle, she waggles her body with her head straight down. So it looks a little bit like this. And you can see that if straight up is 0 degrees, the bee is making the angle of 180 degrees because her body is straight down at 180 degrees from the vertical. In that other situation where the flowers were off to the right, this bee inside would dance with her head straight to the right. So her body would waggle, looking a little bit like this. So that's direction, but what about distance? Bees communicate distance by the duration of the waggle run, which is this portion of the dance where she's moving her body quickly with her head down. So in general, the longer in time she dances, the further in flight she flew. So each second of dance is some unit of distance, but how much? So previously, when people decoded waggle dances, they would use the calibration of Carl von Frisch that's published in his 1967 book to convert duration to distance. But in 2011, when we were just starting some of this work in England, we realized that von Frisch's calibration did not fit our data very well. And this led me and my collaborator, Dr. Roger Shirk, who is also an assistant professor at Virginia Tech with me too. We've had a long-term collaboration. So it led us to do some experiments in England where we were going to calibrate the data. And then more recently, we also did a calibration in Virginia as well. So in these calibration experiments, we set out artificial feeders with a highly rewarding sucrose solution. You can see it here in blue at known distances from the hive. We know how far away these feeders are. This is the undergrad, Kieran Werner, who was working with us that summer. And then we train individually marked bees like this one here to forage at these feeders. And these bees, because they're collecting a highly rewarding sugar solution, are motivated to recruit their sisters to this source. So back in the lab, we have an observation hive, which is a beehive that's a little bit smaller and has glass walls that allows us to see inside the behaviors, including the waggle dance. So that bee with the number tag on her back that was collecting that highly rewarding sucrose solution, she comes back to the hive and she's super excited to tell her sisters about it. So she makes waggle dances and we are ready with our cameras and we film the dances. And because that bee has a number tag and because those feeders are at known distances, we know where she has just been. And so then we can analyze the dance that she does against the distance that we know she has traveled. And then we do that at multiple distances. So here is an overview schematic of where this experiment took place. The H is where the hives are located, and each one of these gray circles is a feeder that we sequentially trained the bees to. So we started at a feeder at 55 meters, and then we get the data there, and then we stepwise our way up to the 622 meters for the 9th feeder. Then we use that data to create a calibration curve. In that particular manuscript, we were creating both a Virginia specific, but also what we're calling a universal calibration for distance to duration conversion. Here on the x-axis, we have distance to feeders. These again are known distances because we've put these feeders out. And then we have on the y-axis the duration of the waggle run, that portion of the dance where she was moving her abdomen in seconds. And we're showing here data from previously published calibrations. So we have 2 data sets that we worked on in England. The red is the data set that we worked on in Virginia. The green is a data set that was published by Adrian Wenner in the 60s. He was a scientist actually trying to disprove the waggle dance, but in the process, he collected and shared some very useful data. And then we took these 3 data sets and we created the universal calibration, which has wider error margins, but we hypothesized would be appropriate for other populations of honeybees or Apis mellifera and from other locations. So some things to note about the universal calibration and the calibrations in general. Variation between bees is really high. So for example, let's look at this little stack of red. These are all honeybees from Virginia in summer of 2018 that foraged at a feeder at the same known distance. And look at the massive spread in duration of the bees communicating the exact same distance. The variation between bees is so high that the variation between sites is not biologically relevant. One thing that's sort of cool is that our universal calibration performed no worse than the site-specific one at recovering distance. So when we decoded these dances, we held out a subset of data, and then we let the universal calibration predict or recover the distance that the bee had communicated, and then we let the Virginia-specific one do the same, and these 2 distances that were recovered were not different from each other. Which is important because some people, we still had some questions about whether or not a universal calibration would be robust enough across different landscapes. So we think that it performs pretty well. It's there, it's available for other researchers. The code and the data and everything are published. And so you can access it if anyone else wants to get into waggle dance decoding, because actually one thing that's really valuable for us is if other groups can externally validate the universal calibration. We've had one such group in Minnesota, the lab of Marla Spivak with her, it was her then PhD student, Morgan Carmarkel. And it looks like the calibration was able to recover distances for them as well. Okay, so this means for our bee who was dancing with her head facing down, she is waggling for approximately 2 seconds. So let's count that. We got 1, 2, and then she stops and turns around. 1, 2, and then she stops and turns around. So if each second of dance is approximately 750 meters, this bee who is dancing for 2 seconds is communicating a distance of 750 plus 750, so about 1.5 kilometers. And if you take these two pieces of information together, right, that the bee danced with her head straight down, making an angle of 180, and that she danced for 2 seconds, communicating a distance of about 1.5 kilometers, you put those two things together and the nestmates inside the hive are able to find the profitable sources of flowers with this information. Okay, so maybe you're thinking, well, that's really cool, Maggie, but how exactly could that be integrated into a research program? Well, for one thing, if we look just at the distance, right, just that component that is communicated with the duration of the waggle run, that has some valuable parts to it on its own. The communicated distance can be a proxy for forage availability. Why is that? Because flight is very costly. When a bee is flying to forage, she is using a lot of energy. And bees are economically savvy foragers, which means they will only fly and recruit as far as necessary. If we consistently see communicated foraging distances that are far away versus another time of year when they're close by, that means when the dances are communicating further away distances that food is less available. We used this previously to examine forage availability in Southwest England. Here we have the different months on the x-axis and communicated foraging distance on the y-axis. And for each month, we took all the dances that we decoded in that month, and you can see the average foraging distance across the 2 years, there was over 5,000 dances. And you can see that the foraging distance varies with season. So in Southern England, where we were located, the bees were communicating the highest distance kind of in high summer, decreased in the autumn. This indicates overwintering. Foraging distances are lowest in the early spring, increase into high summer, decrease again into autumn. So we see a very consistent pattern from year to year. And so for us there in rural England, when the city ecologist approached us and said, we want to make the city of Brighton more bee friendly, we were able to say, well, you should plant something that blooms in high summer because that seems to be the time of year where forage is relatively less available. But that's just the distance component. Let's consider the dance itself. One thing that's very important is that waggle dances provide filtered information. So not every forager dances. It's only the forager who's working the best food at any given time that's going to come back and perform waggle dances. So when we study waggle dances, we're not learning about all the available forage, including the low quality or medium quality, but really only about what the bees are telling as the best at any given time. This means that they provide biologically relevant information about the best forageability and how the bees are currently exploiting the land. And what kind of like scale am I meaning when I say land? To give you an idea, if we place observation hives right here in the middle of this landscape, most bees, will fly kind of around 2 kilometers because distance, again, is a big consideration. Flight is very costly. But bees can fly up to 10 or 12 kilometers. So that means that if a bee is flying 10 or 12 kilometers, the area of land that they're effectively covering and they're scouting can be up to 314 square kilometers. So this is a huge swath of land that it would take an enormous amount of time for an ecologist to actually survey on foot. We let the bees do the hard work. And because they are doing this and reporting back about the the good information in the landscape, the good food, we can ask a variety of types of questions. So I already mentioned that we can look just at distance to tell us when forage is available and how that might change with time of year and with season. But we can also ask spatial dynamics of forage availability. So in particular, the bees are dancing for different parts of the landscape. So we can say, well, where's sort of the hot spots in the landscape, or where's the cold spots in the landscape? So kind of qualitative questions about how the bees are foraging. We can ask more quantitative questions, if we can use with GIS to break down our landscape into different landscape category types, and then we can evaluate how the bees are foraging based on the landscape type. This was a lot of work that I did as a postdoc with Professor Francis Ratniks, which is shown here with his Keep the Hive Alive. And we did this in Southern England, and it seemed to work very well. So our honeybees were located here at this star right in the middle of a large swath of land. You can see that from here, this white line at the bottom is 10 kilometers. We took over 5,000 dances that were decoded across 2 years. We rasterized them and plotted them all at the same time on the map. And we had a nice red hotspot that showed up. And we knew from our local information of the area that this hotspot indicated the Castle Hill National Nature Reserve and Special Area of Conservation, which was an ancient wildflower-rich chalk grassland site, looking a little bit like this. So the honeybees were able to pick out of a big landscape a good spot, and we saw it as a hotspot. More quantitatively, when we divided the landscape up into different landscape categories, these were 7 categories that we obtained from Natural England: urban, rural, and then rural land under different levels of environmental stewardship. We again take all the decoded dances, overlaid them, and then we asked, is high dance activity associated with one of these 7 land type categories? We found that these dancing bees possessed a significant preference for rural lands under the highest level of environmental stewardship. So that's our more qualitative and quantitative data. Another quantitative question that we can ask is percent foraging. So again, you can take the dances happening at a certain time of year, and if you have, for example, a field of interest or a land type of interest, and you can query during that time, what's the percentage of dances that are indicating that particular area versus the other areas, so percent foraging. So this was, again, work that we kind of piloted at the University of Sussex. And one thing that's been really gratifying is in kind of the intervening years, we've had some colleagues that have taken on dancing bees as a way to evaluate the landscape. And these 4 here were all kind of bright-eyed PhD students, are now postdocs or have faculty positions. So Dr. Morgan Carr-Markel used the waggle dance to evaluate how the bees were using reconstituted prairies. Dr. Nick Balfour used the waggle dance to see how honeybees were using a commercial crop fields. Dr. Doug Sponsler using of agricultural land in a choice experiment in Ohio. And Dr. Michal Garbazov was evaluating how bees were foraging on spring flowering oilseed rape. So the dance is obviously very cool and exciting and giving good information to people, which is why it's I've taken it with me in Virginia, and I've used it to start a large 5-year project that began in 2018 that I call Bioindicators for a Sustainable Future, funded by the Foundation for Food and Agricultural Research. So most of what I'm going to talk to you about now will be the data that came from this first phase of the project. In this first phase, we established observation hives in 3 locations across the state of Virginia. So Virginia is on the East Coast in the United States. At each of these 3 locations, we put 3 observation hives, so 9 hives in total. The locations were our Bee Lab and Blacksburg, because this is where the main campus of Virginia Tech is located. In Northern Virginia at the Alson H. Smith Jr. Agricultural Research and Extension Center in Winchester, so we call this Warwick. And then in Southeast Virginia at the Tidewater Agricultural Research and Extension Center that we call Terrick. So we have Warwick, Terrick, and Blacksburg. The Bee Lab is called Price's Fort. Our observation hives were inside sheds or buildings, but tubes led to the outside. So the bees could forage naturally at the landscape, and they returned to their hives through this wall, and then would dance inside the hive. And we would record 4 times a week from April to October. And we did this for 2 years. Concurrently, 2 times a week, while we were filming, we also collected forager return pollen. So the pollen foragers would come back, crawl through this mesh cap. It would knock the pollen off their legs, which collected at the bottom of the tubes, and then they would go inside and proceed to do waggle dances in the observation hive. And we did some cool things with that pollen I'll tell you about later. Then once we had our 2 years of dances, we would upload these videos and decode by hand the dances that we saw. We used ImageJ to recover the angle that the bees were communicating, and we used the start and the stop frame to recover the duration, which then we can convert to distance. We end up with about 3,500 dances per site, and there's 3 sites, so a little over 11,000 dances in total. I'm first going to tell you about some data coming out of TERIC. This is the master's work of my student, Mary Silliman. She graduated in 2021, and she was looking at how honeybees were foraging in this environment, which was a focus of row crop research and extension. So this is an overview. This is our bee shed. You can see the 3 openings for the 3 observation hives. What are row crops? These are things like corn, cotton, peanuts, and soybean, largely self or wind pollinated crops, but crops that produce pollen and sometimes even nectar nonetheless. Because they're wind and self-pollinated, we don't know a lot actually about the foraging dynamics of honeybees that are located in this environment. So our major row crops, the peanut, soybean, corn, and cotton, this shows their bloom time. So April to October, where the color is the darkest is where they are most in bloom. And so where we were, the month of July, all 4 row crops at some point were full bloom. So first, we're going to look at communicated foraging distance, which gives us an idea of food availability. So again, we have distance on the y-axis, and we have month on the x-axis. This is 2018, this is 2019, and a total of almost 3,500 dances. And for our bees located there in TERIC in Southeast Virginia, July in both years had the shortest median communicated distance, which would indicate abundant food nearby. So this was very exciting for us because this is different from what we saw in England, where in England, high summer, July and August was the time when we had the furthest distance. And so it sort of suggests something different was going on in this row crop environment. So then we decoded the dances that happen in the pre-bloom time, in the full bloom time, and in the post-bloom time. We mapped on the landscape where the soybeans, the cotton, the peanut, and the corn were blooming. We overlaid the dances, and we determined percent foraging into our fields of interest. And what we found is that honeybees recruit to peanut, to corn, a little bit to cotton, and not to soybean. How do we see that? So on each one of these graphs, we have percent visitation on the y-axis, and on the x-axis, we have pre, during, and post-bloom. If the bees were increasing their recruitment to these row crops, then we would see the highest recruitment would be during full bloom time. So there should be a down, up, down pattern, which is what we see here in peanut. So say they're going to the fields a little bit during pre-bloom. You know, there's some flowers that are blooming, but during full bloom, they really increase their indication of these fields. And then it decreases again in post-bloom. We see a similar pattern of corn. In cotton, we see a difference per year. So they seem to use the field of cotton in 2019, but not 2018. I can tell you a little bit about that if you're curious. But they do not seem to use soybean. If anything, the recruitment to the soybean field is lowest during full bloom. We analyze this also with odds ratio. In odds ratio, we're statistically correcting for distance, and we can determine that peanut fields in July are the most attractive time and area, as indicated by the honeybee waggle dance. So if you look, the peanut is here, this kind of mustard yellow color, and the 1 is our odds ratio. And so this is the highest time, so in July, peanuts are indicated the most by the waggle dance. Peanuts, of course, are yummy, but in general, we were actually surprised how much overall the honeybees did seem to forage in these row crop fields. So if we took all the dances that were kind of indicating row crop fields in July, it seems that about 50% of all the recruitment happening in July was indicating some row crop field, which is why we're sort of feeling like row crops are providing an all-you-can-eat midsummer buffet, right? Those communicated distances were lowest in July. These bees were located in that environment. The fields are all around them, and they went to them a lot. The next question, of course, is this good? So if you have a never-ending ability to eat at McDonald's, that doesn't necessarily mean that you should. So that's sort of where we're going to now with this research, is to trying to determine a little bit the health consequences of the honeybee's use of the row crop environment. So stay tuned. I'm going to move on quickly to my next location, which is a fruit orchard. This is the masterwork of my student Taylor Steele. She also graduated in 2021. And here is Northern Virginia, Winchester, which we call Warwick, which is focused on fruit crop research and extension. So here we see a little bit of some grapevines, but a majority of Warwick is apple orchards. Where this red barn is right next to where we moved our shed. So here is our bee shed with our 3 openings for the 3 observation hives. And right there in the middle of apple fields, we could, of course, see that the apples bloom mid-April to mid-May. So a fairly predictable and fairly limited bloom time. We hypothesized that our bees located there in the apple orchards would experience a feast and then famine effect, right? So when the apple orchards are blooming, they heavily recruit to the apple orchards. And then once they stop blooming, they have to go, for example, further afield. I'm just going to skip ahead and tell you that is not what we found. And I'm going to build the story up for you sequentially in the way that it was built up for us. So the first clue that something different was going on here was when we looked at, again, distance over time. So distance as a proxy for forage availability. Distance is here on the y-axis, and then we have bloom season on the x-axis. We have during bloom. So the study could not start early enough for pre-bloom because Winchester is fairly chilly, and so we only were able to put out the bees kind of second week of April, and the apple trees were about to start blooming. Or the apple trees had just started blooming. So we have during bloom, we have post bloom, we have late summer, and we have fall. So a quick word on how we delineated these seasons. Bloom is decided for us, right? It's that kind of mid-April to mid-May. It was about 41 days for each year. And so then we said, okay, if that's bloom, we're going to call bloom the 41 days after that period. In Winchester, and actually in a lot of temperate regions, there's often an autumnal forage source that's very important for bees. In our spot in the eastern US, it's goldenrod. In England, it was ivy. And so for us, goldenrod starts to bloom kind of in the fall, and it keeps blooming up till the end of the foraging season, again, about 45 days. So if that's fall, then we said that's 45 days. We're going to call late summer the 45 days before the goldenrod is blooming. So that's our seasons here on the x-axis. If apple blooms were very important to bees, we would see them driving the communicating distance. So we would see the shortest distances kind of in bloom, and then we would see this increase in post bloom, but that's not actually what we saw. If anything, there's not a significant difference between bloom and post bloom in the distances that are communicated. Summer is when we see in Winchester the highest communicated foraging distance with a drop into autumn, huge error bars in 2019. So communicated foraging distance did not increase from bloom to post bloom. So we're like, okay. So then we did our percent foraging calculation where we took the dances that occurred in the bloom and we took the dances that occurred in the post bloom. And we have our 2 years. When we're using GIS, we marked where the apple orchards are, and then we marked where forests are as a control, a contrast, because we don't expect apple blooms to affect recruitment to forests. And we determined the percent foraging in these areas during the bloom and the post-bloom time. So red is apple, black is forest. These dotted horizontal lines are the representation of that land type in our foraging region, which is 2 kilometers from where the hive is. So apple orchards comprise about 16% of the land cover. Forests are comprising about 25% of the land cover. If all things are equal, then you would expect bees to recruit to the apple orchard about 16% and recruit to the forest about 25%. And instead, they do recruit higher than expected during the bloom. But for apples, they increase their recruitment in the post-bloom period for both years. Forests, as we go from bloom to post-bloom, the percent foraging, as indicated by the dance, either stays the same or decreases. So bees are recruiting to apple orchards more in the post-bloom period when the apple trees are no longer blooming. Again, we can look at odds ratio that corrects for distance, and the bees are twice as likely to recruit to apple orchards in June, so here, after the bloom in the post-bloom period, that's June, compared to April or May, which is during the bloom. We can, of course, divide our dances up into dances for nectar versus dances for pollen, because you can see the pollen on the legs. And the important take home from these graphs, again, we have percent foraging on the Y and we have bloom and post bloom on the X, is that this increase in recruitment to the orchards, the apple orchards in the post bloom period is largely driven by pollen foraging. So the pollen here is the dashed line, and you can see that it really increases from bloom to post bloom. Nectar also increases. In this second year, the pollen foraging increases proportionally even more than the nectar foraging. Okay, so this is good because we collected pollen from returning foragers, so we can bring that piece of the story into our puzzle. We did some observational ground truthing in 2020 because we were starting to get a sense that the story was a little different than what we'd originally hypothesized. This is right kind of in the middle part of COVID lockdown, and so my student wasn't able to do ground truthing in the bloom period, but she was able to go in the post-bloom period, and she cataloged the abundance of different plants that she saw in either orchards or forests. And in the post-bloom period, the apple orchards had a profusion of wildflowers growing in the understory of the trees. So, you know, the trees have done blooming, so perhaps the mowing is less of a priority, and we see plantain, red clover, and white clover growing in high abundance in the orchards. So this is correlational, but we had the pollen from the legs, and so we could send the pollen off for some metabar coding, which tells us sometimes down to genus, but often family of the species of plant that the bee had been collecting from, right? This is the pollen that was on their legs. And this is what is in this graph, which is color coded by different plant types. So the kind of two important take homes. During the bloom, you see a lot of red, that's apple, which is good, right? They do go and forage for pollen on the apple trees during the bloom. So a nice kind of a positive control. And in the post-bloom period, we see a profusion of green, which is plantain, and purple, which is clover, which happen to be the most abundant wildflowers and you know, leaf flowery in the understory that my student had seen when she went and did some ground truthing. Okay, so that seemed to suggest to us that yes, honeybees are recruiting to apple orchards, but they're increasing their recruitment in the post-bloom period, and this is what they're foraging on, this weedy, flowery understory in the orchards. We also took some of that pollen and sent it off for pesticides analysis. And here we have pesticide broken down by total cumulative amount of contaminant, and we divided it for insecticide, fungicide, and fungicide and herbicide. And the post-bloom orchard provide the highest exposure to pesticides for foraging honeybees. So here is during the bloom time of 2018 and 2019. This is the post-bloom time for 2018 and 2019. And look how much exposure the honeybees are experiencing during that time. So let's bring it all together. The apple orchards are feeding and contaminating bees during the bloom, but more so after the bloom. So previous investigations seem to focus a lot on sort of the bees, you know, how the orchard is affected by the presence of bees, but we knew less about how the bees are affected by the orchards, especially if they are located in the orchard past the time when the orchards are blooming, right? I mean, like migratory beekeeping, you move the bees in, the orchards are blooming, they do their thing, and then you move them out. Here, we're letting them stay to see what happens. And it seems that the post-bloom period is providing abundant forage because I guess the orchards perhaps are not managed in the same way after the trees have bloomed, but also abundant exposure opportunities. So this has a lot of implications, not just for the reciprocal relationship of the bees to the orchard, but also for management too. Okay, so I'm going to finish up now because I know that we want to leave some time for questions. I'm just going to tell you a very quick thing. So this is Brad Olinger. He is my PhD student. He'll be graduating in this next year, which is super exciting. So Brad's looking at the honeybees that are located here in Blacksburg. I'm going to hold off telling you about some of the things he's finding. And he's also looking across all three sites. So what are some common trends in time and space of the bees that have been in all three of these sites? And additionally, Brad is interested in a lot of kind of more fundamental questions of honeybees, and he uses the dance to evaluate some other things, including the effects of pesticides. So we recently, actually just this year, have published a paper where we took observation hives in a location and trained the bees to forage at two equidistant feeders of artificial sucrose solution. One is a control feeder, and it's 1 molar, and the other one was also 1 molar, but had a field realistic concentration of imidacloprid, which is a neonicotinoid pesticide. And we trained these bees, and then we monitored their foraging and their recruitment. And the honeybees that went to the imidacloprid had about a 28% decrease in their foraging frequency. They saw about a 66% decrease in their persistency the next day for the treated versus the control. So the waggle dance can tell us all kinds of amazing things, you know, like how foraging and recruitment can be affected by a treatment like this, but also how the bees are using different landscapes. Moving forward, we are wrapping up phase 1, trying to get the first papers published and the first set of students graduated. Over the next year and two, we're going to be going into a second phase where we take all that information that the honeybees have told us, and then we're going to experimentally vet it against other non-honeybee bees. So we have 2 additional students. This is Chad Campbell. He's a PhD student in my lab. This is Rob Ostrom. He's a PhD student in my collaborator, Roger Shark's lab. And what Chad will be doing is in those 3 landscapes around Virginia where the honeybees have evaluated the landscape and reported back to us the good and the bad of where they're collecting food and where they're not, he is placing out alfalfa leaf cutting bees. So another managed European bee that's not a honeybee, he's placing out these alfalfa leaf cutting bees, and he'll be tracking the productivity and survival of these bees. And our hypothesis is where the honeybees have indicated things is good or bad will be supported by the health and productivity in the good areas of the alfalfa leaf cutting bees, or their lack of health and productivity in the not so good areas. Meanwhile, Rob Ostrom will be surveying the abundance and diversity of the wild bee population. So using trapping methods, they'll be sampling across these 3 landscapes in Virginia. And again, the hypothesis is where the honeybees have indicated great for forage will also be good for supporting abundant and diverse bee communities. Okay, so to wrap it all together, knowing where an animal collects its food is important for any conservation effort. So if you are a marine biologist, you will work very hard to get this transponder on the back of this whale, and it will collect positional data as the whale feeds. But then, you know, you may have to retrieve this device, which is really difficult. We don't have to do that with the honeybees. Right? The honeybee is the only animal that can tell you where it is foraged, making the waggle dance information unique and biologically relevant and one of a kind. There is no other way to get these data. The only animal that can tell us where it is eating. So it would do us all well, perhaps, to listen to that. You're welcome to tune in for future updates. So I am one of several bee assistant professors at Virginia Tech. We have the bee group at VT with a Facebook page. You can like us. We don't post too often, but we keep you updated about some research that's going on. My own lab has a website, and I am an infrequent Twitter user, but I'm trying to get better, and that's my handle. And lastly, I'd like to thank, firstly, all of you for being the audience of this inaugural Frontiers in Bee Behavior and Ecology talk. But I'd especially like to thank our organizers, so Drs. Christoph Ruther and Axel Brockman, for the invitation. In the first place, I'd like to thank the members of my lab, past and present at Virginia Tech, and this research is supported by a NIFA, there's the project number to me, and a donation from FFAR and a donation from Kaiser, or sorry, grant from FFAR and a donation from Kaiser Compressors. With that, I am happy to take any questions. And thank you very much.
Thank you very much, Maggie. That was a very clear and very interesting talk. So as a reminder, you can, if you have questions, you can either put the question in the chat or you can raise your virtual hand and ask the question directly. I think Axel will look for questions in the chat.
Yeah. There are several questions in the chat. So the first question is by Vidur Niranjan, and it's concerning the universal curve. Am I audible? Has anyone tested African or Africanized bees and compared those with the tentative universal curve? That means kind of, Maggie, does your universal curve hold for other populations of Apis mellifera?
No, we have not, and I would love to have someone do that. So if anyone is interested, please contact me.
Okay, good. Good answer. Okay, the next question is by Chris Kreusling. Question, do measurements of foraging and recruitment increase because the colony size has increased during the year? Or are those measurements scaled to the hive size at the time of measurements?
Yeah, that's a great question. So observation hives have some pros and cons, but the end result is that we have to maintain them at a really set size. They're not allowed to grow, the cavity space is small. They're not allowed to grow beyond it because they would swarm and then we lose our experimental bees. And we also maintained our hives for consistent size throughout the entire experiment for this very reason. So this seems to be irrespective of colony size for these data.
Okay, good. So Natasha Rossi, thank you for your very nice presentation. Previous studies have found that workers following dancers' instructions actually represent a minority in the colony. Most workers seem to be reactivated to forage using private information. That means their own memory. Do you take this could have an impact on your results?
Yeah, so that's a great question, and kind of getting at more of the fundamental nature of the waggle dance. So for the purposes of our study, we're not actually interested in the recruits, because we're tracking the behavior of the dancers. So the dancers are telling us where they have been, and then we use that to make our calculations for the use of the landscape. But it's the dancers' use of the landscape, not the recruits. But you're correct. The dance is famously, well, as you said, the bees themselves, of course, are heavily influenced by their private memories, their memories of forage location. And then also the dance itself is famously inefficient, in that even when bees follow the dance, not all the bees are successful in finding it. But luckily for our study, their behavior is less relevant.
Okay, then there's a question from Pierre Lau. Question, do you have any information on the soybean variety that were present in your raw crop study, and if they were nectar producing types?
Oh, hey, Pierre, that is a great question. So we would know the variety because my collaborator on that work is an entomologist that's based there in Terek, and she is a row crop entomologist. So if you would like that information, I can get it for you. It'll also be in a paper that hopefully will be published soon. I can also hook you up with Dr. Taylor. Her name is Dr. Sally Taylor. And they were nectar producing. There were some extra floral nectaries for it.
Okay, so then there's a question by Alberto. Is there any link between wild pollinator biodiversity in orchids and their food production?
That's also a very good question. So there does seem to be a positive association between kind of the bee community that's naturally present. And there is some evidence that suggests that it's their presence that actually has a bigger impact on fruit set, not the arrival of the migratory honeybees that come in. There are, I should say, there are also some studies that seem to show that honeybees do increase the fruit set. It could be that this apparent contradiction is because of just numbers. You know, honeybee hives have thousands and thousands and thousands of workers, whereas kind of like an osmia colony will not have so many. So by sheer number alone, it seems the honeybees are doing some work, but it does seem that it's largely the wild bees that are probably per unit the most efficient of the pollinators.
Okay, then there's a question from Mamta Bardade. How to study bee flora preferences and foraging efficiency in different areas? So very general question.
Yeah, that's a great question, because of course we only did this in 3 areas, so that limits a little bit how broad we can make inferences across the whole world, right? I mean, we put our bees in these 3 landscapes. I think in general, honeybees are going to go to where they can find abundant resources that aren't too far away. And this can change with season, this can change with rainfall, this can change with the available flora, fauna that's in the area. And that seems to be over and over what we're coming back to, that, you know, there needs to be kind of an abundance of different things available throughout the foraging season. This is one way to study it, where we let the bees go and scout the landscape and come back and dance, and then we can mark where that dance is indicating on the landscape. There's other ways too. People have often done trapping methods to survey where the bees are, because of course many other wild bees have shorter foraging ranges, and so where you find them on flowers tends to be where they're nearby where they're living as well. And there's a variety of methods too. You could also look to see which plants are having kind of successful pollination events, and you can infer from there that pollinators are present. So there's different ways that we can look at kind of general foraging biology. This happens to be a cool one because the dance is just really charismatic and fascinating for its own right. I hope that answered your question. If you have any follow-ups, you're welcome to get in touch with me.
Okay, so then there's a question by Dr. Butt. Nice presentation. Is there any difference in dancing by pollen foragers and nectar foragers? That's the first question. The second question, is there any correlation you have done between foraging activity on the crop and dance recruitment during the bloom and post-bloom period?
So let me answer I'm sorry?
No, go ahead, sorry.
So the first question, there doesn't seem to be any differences other than we can see the pollen and the corbiculae of the pollen foragers. So you can see the pollen right there on the legs, and we can identify them as the pollen versus non-pollen dancers, presumably nectar, but the dance itself is the same. Your second question, was it evaluating kind of dances versus their use on the crop, foraging in the crop? So unfortunately, we don't have any information on that for 2 reasons. One is, Tarik is about a 7-hour drive one way from Blacksburg, and the students and I are located in Blacksburg. So we had to depend on the efforts of our collaborators and a nice beekeeper in Winchester named Rusty Foltz to turn on and off our cameras. So we were not there physically during the study because we filmed 4 days a week from April to October. The second thing, though, is that we, of course, decode the dances after the fact. So these data take several years to get. And so we're not getting real-time data. To get that, what you really need is to have an automated way to decode dances in situ and then be able to go out and check the landscape. And that should be a good advertisement for one of your future speakers, Tim Landgraf. So I hope that he'll be talking a little bit about automated methods of dance decoding. We are still doing it by hand.
So then, okay, so I think that's a question to me and Christoph. Martin Navrot, great talk. Will this be available online? So yeah, it will be available online. And Christoph, we hope that we will do it soon on YouTube, and we will let the people know.
Yeah.
So, I mean, there are a lot of people kind of thanking you for the talk and congratulating you for the talk. I skipped that, yeah. I can count it, kind of like.
That's okay. Thank you. It's nice to know.
So then there's a question from Jahan V. Tiwari. I'm sorry, even kind of, I have problems pronouncing Indian names, although I've lived here in India for a long time. Okay, so a question is, what are your views on impact of managed bees on native pollinators?
Oh, that is a hard question. Because I, of course, did my training in England, and so this was less of an issue. And the honeybee is and will always be my first love. I think they are just a fantastic organism. I don't really know. I think it probably depends on the landscape. I think it depends on the density of hives. I think it depends on how, managed or unmanaged the foraging resources are. There's some evidence that suggests there's a synergy between the two, but there is, of course, plenty of evidence that suggests that wild bee diversity decreases with density of hives. So I guess it depends. I do think that for their numbers and their economic impact alone, honeybees are not going to go away. I think future management decisions going forward need to take wild bees into account, though. And probably the most efficient and economically impactful and sustainable management will need to consider both. And we'll need to know, the unique biologies of the wild bees versus the honeybees, and utilize the strengths of each without maximizing on the weaknesses. So I'm going to sidestep it a little bit to say I do think that there is a negative consequence of having really high densities of honeybees. I don't think that's always the case, though.
Okay, so there's a question by Morgan Carmichael. For the comparison with other bee species, will you be looking at pollen, bracket, in comparison with what honeybees collected against areas they advertised, or focusing on health of the leaf cutter bees and abundance of other bees? Was it clear?
Yes, it is. Great question. So I think what it's saying, or I'm going to hope this is what it's saying because I have an answer to it. So where we're putting out the alfalfa leaf cutting bees, the founders is provision for their next generation. So there's pollen that's packed into the nests, and of course we can collect these nests and take the pollen and sample it. And similar to what we did with the honeybees, our hope would be to send it away for the metabar coating. I don't at the moment have funds for it, but we're currently applying for funds. But we have that pollen that we're collecting back. And it would be very interesting, since we have the pollen data from the honeybees, to be able to compare it to what the alfalfa leaf cutting bees are bringing back. So my PhD student, Chad, additionally is doing ground floral surveys. So this time we're doing it a little bit more officially. We have on his PhD committee a professor who has more experience with this, and he'll be helping us out. So we hope to, yes, create a picture of what is what flowers are contributing to the diet of the alfalfa leaf cutting bee.
Okay, so okay, let me make a kind of a general statement. I think now, because we allowed people to put the messages in the chat box, everyone is doing it. Everyone is shying away to raise their hand and ask the question personally. Maybe kind of next time we should do that. Otherwise, you're just hearing my voice, and it might be more interactive. Okay, but I'm going ahead. So now the next question is Julia Mainardi. Great talk, thanks. Did you find a correlation between the pollen metabar coding quantitative results and the amount of dances for specific plant species crops?
Oh, well, the dance is neither, it's inaccurate and imprecise. And so it would be, with the exception of what we hoped would be these orchards or these row crops, you would have a hard time saying the bee is going to this plant, right? We can, I often get asked, how certain are you that's where the bee is indicating? It's like, well, we know that's what the bee is indicating in that area, but it's kind of plus or minus 150 meters. So and I thought that would be foolproof in the orchard. I thought, right, yeah, the orchard is just the apple trees that are blooming, but this is again that was my naivety to not realize kind of how the presence of an understory would impact our results. But even within that, we can only really say the bees are going in general to this area. Now, because we decode so many dances, it allows us to have a better estimate of where the bees are going, right? If each dance is a little imprecise, but we have a lot of them and we rasterize them together and plot them together, it lets us have these kind of hot and cold spots, but it's beyond the resolution of these types of data to be able to point to a specific plant.
Okay, now welcome Gart Otis, friend of mine. Okay, wait a minute, what is happening? Okay, sorry. Okay, so Gart Otis is asking, there was a huge variability in the results of several of your studies between years. How do you address that for coming to generalizations?
Yeah, it's a difficult question and a difficult issue because of course we get the data several years after we collected the data, which limits a little bit some of our ability to collect covariate data. Some things we can recover after the fact. So for example, in Winchester, we saw huge error bars in 2019 that we didn't see in 2018. And in 2019, we had a pretty bad drought in the late summer and the early fall. So usually September sees 4 inches of rain in Winchester, and that year it got like 0.4 inches. So a huge magnitude of difference. And so I think kind of, perhaps on the days when it rained, the bees were able to forage nearby and then they had to go far away. So there did seem to be yearly effects. And then another thing that we saw with Tidewater, for example, the cotton. So honeybees really used the cotton in 2019, but didn't use the cotton in 2018. And again, there was a drought in 2019, and cotton is more drought tolerant and can extend kind of its bloom time. So it could be because it's more drought tolerant, it was able to continue to produce forage while maybe some of the other crops were suffering a bit. So the honeybees increased their recruitment to it is one of our kind of explanations of what's going on. But that is a limitation of the data of only doing 2 years. And also, you only look at the data years later, and then you're trying to recreate using weather data and kind of local on the ground knowledge what had happened in 2018 and 2019. So it is difficult, and so we have to be very careful with kind of what our take homes are from these messages because of that reason.
Okay, then there's a question by Carmen Niebauer. Thank you for your great talk. Questions for the found pesticides in the post-bloom orchards. Do you know how many different pesticides were present? That means 5 different herbicides, 3 different fungicides?
Yes, let me see if I actually have that. Did I have a slide with that? I don't have a slide, but we do have that data. We have quantitative, I mean, we kind of, so that's a very simplistic graph where we just sort of added up what happened, what was in the different time periods. But we do have the specific pesticides and their amounts that were brought back. And of course, the samples, we knew the dates that the bees recovered, this pollen sample came from this date, this pollen sample came from that date. We have all that information, so we can, of course, correlate it with what we know was being sprayed at the time. So we do have that information. It's a massive mountain that I haven't yet started to scale. I'll need to recruit people who have expertise in sort of toxicology that I don't currently have. But we do hope to kind of be able to chip away a little bit at that, because it seems that there's a really interesting story there that, like this post-bloom orchard feeds and contaminates bees. Yeah, we need to know a little bit more of the details. So stay tuned. It'll be at least another year, but that data are there and we will be looking at it.
Okay, then there's a question by Peng Tianfai. Thanks for your report, very interesting. Do you test the levels of chemicals in pollen collected by bees in orchards post-bloom? Could it be that bees visit more during post-bloom and not because they like the plants, but because of the irritation from pesticides?
Irritation from pesticides?
Yeah. So could it be that, okay, wait a minute, I'm jumping. Could it be that bees visit more during the post-bloom and not because they like the plants, but because of the irritation from pesticides?
Well, they do visit the orchards more in the post bloom, and they are coming in contact with more contaminants, pesticides, herbicides, and fungicides during the post bloom. So if there is an irritation, it doesn't seem to be deterring them from indicating the orchards, because that's when percent foraging is highest. If I'm not understanding that correctly, please jump in.
I'm sorry, I'm also not really understanding that. Peng Tianfai, I mean, you can raise your hand. I mean, you kind of like, you can speak to clarify your question. Are you still there?
Yes. Can you hear me now?
Yes.
Yeah.
Yes. Because the pesticide can induce the bees visit the plants more. So I want to ask, because maybe they do not like this plant. So just because of the pesticide induce them to. Yeah. That's my question. Yeah.
Okay, so you were referring to there's been some, if I understand correctly, there's been some reports, most notably the 2015 paper Kessler et al. That was published in Nature, that seemed to suggest that honeybees and bumblebees can increase their foraging in the presence of certain insecticides, certain pesticides. It causes, it's almost like an addiction, and they forage and recruit more heavily. But of course, we also recently did a study that seemed to suggest in a kind of a cine field condition that honeybees decrease their foraging in the presence of neonics. So for that specific issue, I feel like the jury are still out. It probably depends on the landscape and the overall forage that is available for the bees, if there are alternatives. The Kessler paper was a lab-based study where the bees were choosing between Petri dishes. So very valuable data in that paper. I don't know how it would translate on a landscape scale. And additionally, I don't know if that's what's happening in our particular landscape. There's the possibility that some of these pesticides could have a pharmacologically active component that is influencing the foraging behavior of bees. That is possible. But I don't think all of them would have that same effect. And we would have to look into it a little bit more carefully. But we do know that clover and plantain is attractive forage for bees. So it's not really surprising that the bees would go to white, red clover and plantain, plantago.
Yeah, thank you for your answer. Thank you. Yeah.
Sure.
Okay,
there's been some great questions. It's a lot of fun.
Okay, so there's one by Riaz Hossein. He said, that's not a question, it's kind of a request. Kindly share the publications. I think, can one get your publications on your webpage? I mean, so it's probably, I mean, this is a problem here in Asia, right? If people don't have access, but through the publications.
So for the neonic paper is published and it's open access in Journal of Insect Science. The two papers, the first papers for both the row crop and the apple orchard are submitted, and both of them are submitted to open access journals. So we're really trying to target those. Both of them are currently in review, and when they're published, stay tuned. I hope they'll be available. I'm always happy. I always send reprints whenever someone writes me. So my email address is available. You're welcome to write if there's something that you can't get. It'll probably be a little while before the pesticides data are published. That one is only in prep at the moment.
Okay, then there's an interesting question, also again by Dr. Butt. Can the bees communicate pesticide fungicide application on host flora? I mean, or one has to ask it kind of, does it affect the communication? And
right, well, when we did our neonic study, we were hoping to kind of get a little bit a clearer answer on that. There does seem to be some effect on foraging and recruitment behavior. We showed a decrease in foraging frequency. We did show also a decrease in waggle dance frequency and waggle dance propensity. It was non-significant, but in trending in the direction we would suspect based on the foraging data. And there's some other studies that have come out of some other labs. James Nye at the University of California that has looked a little bit more into what are the impacts of pesticides on different aspects of the dance. I don't know if a dancing bee is able to somehow communicate to her sisters that she is dancing for a contaminated resource. I mean, if she can trophylax, but that depends so many things. Can they taste the contaminant? Are there taste receptors for that contaminant? I don't know. If a bee is dancing, by definition, she is evaluating that resource as good. And so I feel like any dance that we see is per definition for good. So whether or not it is actually good, that's a different question, but the bee thinks it is, or she would not dance.
Yeah, so that was, okay, let me check. Okay, so then there's a follow-up question by Carmen Neubauer. Small follow-up to my pesticide question. Do you have data for both pollen and nectar content? Because in most papers, there is a focus on just one of those and not both.
Right, we only have pollen because again, we were located kind of far away. So we collected, it's easy to collect pollen from returning foragers. It brushes, it comes off of their legs as they enter the hive with these pollen traps. To collect nectar, you would have to get a returning forager and chill her and then palpitate her abdomen to get her to regurgitate it, which decreases the likelihood that she'll then dance. And you also then physically have to be, there on the ground. So we were relying again on the kind of the help and work of volunteers and students and beekeepers. And I felt okay asking them to put a pollen trap on the entrance and collect the pollen, but I wasn't able to kind of get them to collect the bees, and then we would lose all of our dance data. So, for us, the pollen data is going to be a really interesting chapter, but I am first and foremost kind of a bee foraging biologist. And so the dance was sort of the main data set, and the pollen was kind of adding the pretty trim. It ended up being a bigger trim than I thought. So unfortunately we don't, but that would be a really interesting thing for future studies to do. So if anyone out there is bright and eager, wants to take it on and has the resources, it is possible to of course evaluate the nectar as well.
Okay, and then there's a question by Pamela Borges. Thank you for the amazing presentation. Do you think with this study will be possible to analyze how many times the same bee can foraging the same area, using the dance to analyze specific behaviors of individuals? So it's a little bit cryptic.
Right. With our bees were not individually marked in the main study. We just set up cameras and we video recorded the dances that were already occurring. Without individual marks, it would be impossible to be able to identify that this dance came from this individual bee. So to do that kind of data, you're talking about a good amount of work. You need to sort of have the entire colony marked, let them go out and do their thing, and then have probably ways to automate first the identification of the bees. I mean, it can be done. There's some great work with Gene Robinson's lab where they're using different tags that are on the back. And then to be able to link individual dance information with the tag. I mean, also you'd want the tag to not impede the dance. It has to be small and readable. But I think they could probably do that in the Robinson lab. And then to be able to determine that this bee has gone to this place. So for the purposes of our study, that was beyond the scope of what we could do, beyond the scope of what I guess we were looking to do, because we're depending on large numbers, right? We need lots of dances. But that would be a lot of fun to have a system set up to do that.
It would be interesting also with the radar
Right.
method by Randall Manser. But
Right.
how to say, there's a problem that they have to remove the sender, right? So,
Right.
but I think there might be some data that they show flight behavior of individual bees on consecutive days or so.
Yes.
I mean, if you want to analyze the variation, kind of,
right.
I mean, I think that we would think that the bees are going basically to the same area. Now the question is, what is the area, right? And how they explore the area from foraging to foraging trip.
I mean, we do, you know, the bees, when they have learned a valuable resource, they are very committed to that resource. And we know that from some good work that Christoph did as well. You know, the honeybees who have had experience of a reward, they will stick with that, even if the reward becomes not rewarding. So I would imagine that for, you know, and we see this with a lot of our feeder studies as well. You know, we go out the next morning to set up the feeder and the bees are waiting for us. Like they're buzzing around our head, waiting for us, our slow human bodies to put out this feeder for them. So, you know, with there are other ways, Axel, like you were saying, you know, with the radar, the difficulty is it would have to be kind of an open air hive, right? For a bee with a little antennae to be able to dance. So there's some beautiful studies looking at the flight orientation behavior and the following subsequent days, but that hasn't been linked to dance yet. That would be really neat to be able to do that.
Okay, then there's a question by D. Treshock, and that is also more methodology. Are there pictures of your observation hives? Probably it means kind of as a blueprint for how to build an observation.
So I'm trying to think if we have, so I showed, this is, let me see, can I share my screen again? Is it still sharing my screen?
Yeah, it's shared.
Okay. So this is an observation hive that we have in our bee lab, and it's very similar to what I used in my postdoc with Francis Ratnick's. It's a 3-frame. These are American standard frames, wooden. There's a tube here that leads to the outside. We have here, I don't know if you can see that, that's an upside down mason jar, which when we need to, we feed the bees. We, of course, in our studies across Virginia, we try to restrict feeding to after the data collection are done for the week, but observation hives are notoriously finicky because we're maintaining the bees in a size they don't like. It's too small for them, and we don't let them keep big honey stores. So we sometimes do have to feed. So this is kind of the general plan that we have. And I think so Roger Shirk, who's here, the first author on this paper that was published, he's also an assistant professor. I should say he's also my husband. So he modified the design that we used as postdocs, and he came up with this. And I believe he actually has as a PDF some blueprints for it. And I'm sure he would be happy to share that if that can be helpful. And it has the size and the specifications for it.
Okay, good. So I think that was the last question, but I have a question, and it's more like confirmation. Did I understood it right, that the dance analysis that you are doing is basically by hand? So the student is identifying the start and the stop, and then kind of counting, or kind of like Excel is counting the frames, and then they have to measure the orientation.
So you're correct. We are doing it by hand. It's not quite as bad as it sounds. So now what we do is we're using the application ImageJ, where we play the videos. We identified the start frame and the stop frame of the waggle run, and we enter that into Excel, and then it automatically calculates for us the duration. And then we have our calibration in there, so then it automatically converts it to distance. In terms of the angle, the bee is dancing, and there's a straight edge tool in the ImageJ. So as she dances, we, can draw the line. So she's dancing, and we draw the line that best fits as she moves her body. And then ImageJ calculates for us what that angle is, and then that also goes into Excel. And then we have code that can put the two together, and that gives you the vector.
Okay.
There are some really cool work doing to try to automate this. It's not yet what we need for this type of study. But I'm hoping now that, we got this data set, someone else is welcome to crack that code. I am not a computer scientist, but I would love to have someone find a way to make this easier.
Everyone would like to have that. And
Yeah.
so that is why I'm asking, right? And,
Okay.
and probably it is very important. And yeah, okay, good. I just wanted to know whether you have a better solution than me. And one has to kind of state that, how to say, then analyzing 3,000 or what number of 11,000 is hard work, right? Yes. So kind of like you have to thank your students. I mean, I'm always kind of a little bit shy to ask people to do that.
Yes, there's a huge student component in this work. So wonderful grad students, also undergrads that have worked with my lab over the past couple of years. So this is Taylor Steele. She was a grad student that worked in the apple orchards. But even kind of the image J is a step up. In England, we were measuring the angles with protractors. Like we had an acetate sheet that I taped over the screen, and as the bee danced, I would draw the line and then I would measure it with a protractor. So this is at least a little bit quicker than that.
Okay, then there's a direct message to me, but actually I cannot answer that. So it's kind of like, it's not from Pamela Borges. Mr. Axel, could you please send me the name of Professor Rob that Mrs. Margaret mentioned about my question? I would like to read the publication.
Of which one?
That was Pamela, and so you must have mentioned a professor named Rob.
Rob.
And, okay, I have to figure out again. Maybe Pamela, can you send me another chat message? What was it about? It was, let me see.
I mean, we had here, this student is named Rob Ostrom, and he is surveying the abundance and diversity of the wild bees in the areas that the honeybees live in.
No. So Pamela had asked whether, so then it was Jean Robinson. It was a question regarding kind of whether bees fly to the same area for how long, right?
Oh, okay.
So, and I mean, to follow up, as you said, that actually the foraging behavior of honeybees is social, right? And then there is a division of labor between scouts and recruits tend to stay, actually, as you said, with the location. And we think that a recruit is maybe, I don't know, twice or thrice changing her location.
We think the recruit is, I'm sorry, can you say that again?
No, I'm saying that so the recruits are staying, as you said, with the food source, and only will leave it if the food source is not good enough anymore. And then
even then they sometimes stick around a little bit.
Yeah, okay, true. So they are really kind of, in some sense, because it's division of labor, so they stay with, right? So they are not that variable, but this is because of the social organization.
Correct. Yes.
Like
yeah.
Okay, then I can okay, so Pamela sent me an email, and then I will let you know the publication. Okay.
Then we have a call for action to do more research in Asia on honeybee species, which I think is a very good idea.
Yes.
And read it, kind of like, let's check the change. You read it now.
So we know almost nothing. That was Garth Otis. We know almost nothing about the foraging dynamics of co-occurring honeybee species in Asia. My plea to everyone, extend your research to Asia. And Asian scientists, honeybee ecology needs your attention. I completely agree with that, and would also like to mention stingless bees that also could be studied more in Asia.
Sounds like some wonderful opportunities for travel and collaboration.
It is, I mean, but this is also one goal of this talk. I mean, for different reasons, studies on honeybees and bees in general, and on insects in Asia is low, right? And so kind of make the point that if you talk to Indians here about wild animals, they think about tigers, elephants, and rhinos, which is okay. But insects don't go under the term wild animals or interesting animals. And so we have a wildlife master program, and no one is studying insects there. It's actually the whole research needs to be increased, so I would say that we would need foreigners to help, but it's important that the research in India or in Asia needs to be built up. So it's kind of like more educational. This I'm saying, it's a topic to my heart. This I'm saying because many of the research that has been done so far, I mean, most of good research is also done by Westerners, but it is that the Westerners are getting contact with researchers here, and then they are doing the studies, but they don't really educate. And so I think, I mean, there are not many bee labs in India, and India is a continent, right? And then kind of one has the difficulties, of course, these are also, most of them are wild honeybees. But okay, now I'm starting to talk till 12 o'clock. I mean, I think Christoph agrees that there's a time that we have to finish, no?
I just wanted to ask you, Maggie, I would have a question, but I understand, of course, that you have a life and other things going on.
No, I would love to hear your question.
And maybe then we can wrap it up after this. I'm very interested by this individual variation that you mentioned at the beginning, that bees are collecting food at the same distance, and you see these differences within hive, and also there's some variation between studies. And I was just wondering, what do you think causes this variation? Because I guess there could be different sources. There could be the behavior that is different in that they fly at a different distance, and therefore the visual input is different. It could be that they fly, I don't know, maybe the visual input is more variable in certain environments, or it could be that the brain processes the visual information in a different way. And I was just wondering, do you have any idea what causes this variation? Maybe all of these things.
Right. I don't. I have sort of my favorite pet hypothesis. So I want to just quickly kind of reiterate, yeah, we see massive individual variation in calibration. Not so much, at least in our studies, not so much between hives. It's so high between bees that the hive effect is negligible. The location effect is negligible. Even the decade effect is actually negligible because the individual
She's
variation
doing an
is really high.
international star.
But I would say that kind of within a bee, so that the kind of intra-bee variation is not. And that's why we are able to have these lines. So when we train the same bee to successive feeder distances, you can see sort of her dances, they cluster very tightly for each distance. So there does seem to be this individual component. And my pet hypothesis is that it's genetically driven, that the multiple matings that happen in the hive are producing patrolines that have individual calibration, and that's maintained in the behaviors that we see. But we did recently have a really interesting conversations with a professor named Andrew Straw, who studies kind of the issue of height and like landscape flight to different height, which is something I never considered. So it would be, I guess, not quite as conservative an explanation, but if different, if a bee always tends to fly at a particular height, then maybe that would impact her individual calibration. But there would have to be, yes, some sort of conserved mechanism that kind of allows this to shake out at the colony level and give us what we see as this fairly stable individual variation. I do have a graduate student, Laura McHenry, who this summer is going to be hopefully doing a follow-up work. We have a 2016 paper that looks at individual calibrations, and she's going to be doing a follow-up asking if when recruitment is successful, is it, or should I say, when recruitment is not successful, is it because the individual calibrations of the dancer and the recruit differ enough? So the slopes seem to be different, but the slopes are similar, but the intercepts are different. And the hypothesis is that failure to recruit is predicted by differences in individual calibration. So that doesn't get, again, at the mechanism, but we can collect those bees, which we will be doing. And if we find a very talented molecular collaborator who can do the genetics for us, then at least we could either reject or fail to reject that hypothesis that it has something to do with sort of, but again, if it is genetics, I still don't even know like the mechanism of how that shakes out to the eye.
There's an interesting suggestion in the chat to use the tunnel to compare waggle run durations, because you then would hope you would remove a lot of these environmental variations.
Correct. Yeah, my grad student Brad Olinger, he had hopes of doing that kind of in these summers, and COVID just sort of threw everything out of the window. But one day we'll try to get tunnels up and running here.
I wanted to make a comment. I'm not sure, but I think there might be a publication, maybe by Ben Aldroyd, about dancing bees, or that Patry line kind of like bees dance more often with each other or something like that. I have to check that. So
there might have been also one with Stan Schneider a little while ago, but maybe Ben as well. But I do sort of remember that kind of dancers and dance followers tend to have similar Patry lines, which would be nice. But that so these are all kind of interesting things. I mean, I would. I can't wait to see what happens this summer.
So it was Stan Schneider, you think? I have to check that.
There was he had an undergrad student who then later went on and did her PhD with Anna Dornhaus. Her name was Nee Kao, and I think Nee looked at sort of waggle dances and calibrations, but I don't remember the details of the study.
There is a Ben Aldroyd study as well, where they show that they dance with their super sisters.
Their super sisters.
This is something that could, of course, be driven by other factors, such as preferences for certain types of food and an attraction through the odor and so on. But it's very interesting.
But how they say it would this would have the same come out, right? I mean, if they did it together because of different reasons, but then they align basically calibration curves, right? So, interesting.
Okay, Maggie. Sorry. No, please go ahead.
That's great.
I don't want to cut you off. Well, thank you very much for this really very interesting talk. Thank you to all the participants and the people who ask questions. I hope we'll see many of you again next week, where we have another talk by Andres Arenas. So have a good day and hopefully see you next week.
Have a good day, everyone. Thank you again for the invite.